Get started. Um, Even though I know you guys, you may not know what I do. I'm a professor in the medicine department in the Jerry Grantham Kidney Institute at the University of Kansas Medical Center, and I'm representing the cell model subcommittee. I'm one of the co-chairs for that subcommittee. And the our mission for the subcommittee is to generate and distribute relevant and reliable PKD cell models that can be used to investigate mechanisms of renal cystic disease for target identification. Target identification would be trying to identify a component of the pathway that might be a target for therapy. And then also for testing drugs in a controlled experimental environment. So I think you're all familiar with the kidney and cyst formation, but I just want to point out a couple of things. The cysts form from the tubule epithelial cells, right? So the kidneys are made up of about 1 million nephrons. The nephrons are basically a tube that has a filter on it, and the tubules reabsorb the fluid and control the release of certain ions from the body and get rid of toxins. And that's this shown over here, a cross section through tubules that from a PKD patient. And of course, all the cells have a germline mutation, but something goes wrong with these tubule epithelial cells and makes them take on a different phenotype, and they begin to proliferate and they form a cyst. And in the process, they release a lot of factors into the space adjacent to the cyst that cause inflammation and ultimately fibrosis. And then most of these structures that form from these tubule epithelial cells become isolated sacs. And these epithelial cells, now the cystic epithelial cells or cyst lining cells, continue to proliferate, and they have active mechanisms to drive fluid accumulation within the cyst. And this is an active process that involves chloride transport, much like the airway. It involves CFTR, it's the same gene that's mutated in cystic fibrosis, and those patients can't secrete fluid into their airway. So a lot of these mechanisms that are involved in fluid accumulation in the cysts have similarities to other secretory tissues. Now, the reason I bring this up is that we use these cells to study characteristics of polycystic kidney disease. And these are either cell models, or we also use the term in vitro models. In vitro means outside the body. Last time you guys met with Stephen Parnell, and he talked about animal models and in vivo type of assays that they can do. So the thing about the cell assays is that they replicate the in vivo characteristics, but while providing a very controlled and standardized environment that enable investigators to test hypotheses and test therapies. He talked about doing genetically modifications to the animals to study particular pathways, but cultured cells are much easier to manipulate than animal models. So a lot of times we'll start off doing some genetic alterations in cells and look to see if we actually affect a pathway that we think could be involved in cystic disease. In vitro models are also more cost effective than using drug studies than animal models. And we can test multiple compounds to determine a lead compound that may have therapeutic activity, and also determine dose, toxicity, off-target effects before actually testing the compound in animals. And I hope you guys will interrupt me if you have any questions or if you guys would like to make any comments. I certainly don't want it to just make this into a lecture. A lot of times, a lot of these cell models that we use are cells derived from animals, but a lot of investigators want to test their, oh, go ahead, Lisa, I'm sorry.
No, I just wanted to make the point. This whole approach has gained a certain recent urgency with the NIH really sort of casting a somewhat jaundiced eye toward the use of animal models. And so using in vitro models, particularly from human patients and normal controls, is going to be a really important part of our armamentarium going forward, so it solidifies its importance going forward, and certainly for the short term, given some of the misgivings that are being expressed in Washington. So I just wanted to emphasize these models, while as Darren's pointing out, have their limitations, particularly in the current climate. They're really really important.
Thank you, Lisa. Yeah, so a lot of investigators want to use the human PKD cells to kind of validate what they find in animal cells or in animal models. And this is considered to be kind of translational research, and meaning that you can translate what you find in animals into human cells. And that seems to be important for a lot of pharmaceutical companies. I get contacted by a lot of companies that want to use human cells to determine if the compound that they're interested in or the target that they're interested in is also a good target for in human cells or tissues. So, I think using human cells in vitro models is a really good approach for initial experimentation, but they certainly have limitations in that they don't replicate the complexity of an in vivo type of system. And we'll talk a little bit more about that. So this is kind of what my overview is for what I want to talk about. I'm going to first start off with talking a little bit about the human ADPKD and normal human kidney biomaterials that we provide. I know that this is more in the clinical subcommittee, but a lot of times the cells that we generate from the humans are at the same time that we're generating these biomaterials. So I thought I'd talk a little bit about that. I want to touch on the generation and characterization of immortalized isogenic PKD and normal human kidney cells from both mice and humans. I know that that may not exactly make sense just yet, and I'll talk about what those terms refer to when I get to that section. And then I want to just briefly talk about the PKD pluripotent stem cells that we obtain from patients, and they're used for experiments involving kidney-like organoids. So imagine at some point, a lot of you've heard of the term organoids, but I'll kind of characterize that a little bit more in a minute. Okay, so patients with PKD can donate their kidneys for research at the time of transplant. This is something that we've been doing for a long time. I actually work with the PKD Foundation and help them establish a tissue donation program where patients can contact the foundation, and then we can make arrangements for kidneys to be shipped from their hospital to our core facility. And then when we receive these kidneys, the tissues can be isolated and fixed in formalin or paraformaldehyde. That just mainly fixes the tissue so that it doesn't break down. And then we can embed that tissue in paraffin wax for histology sections. And down below here, I've shown what these sections might look like from a normal human kidney and from a kidney from a PKD patient. And you can see these over on the left, these tubule structures. Maybe here, is my pointer working here? Okay. So these are the tubules that filter and reabsorb the fluid that before it becomes urine. And then over on the right is a PKD kidney. And this space up here is all cyst fluid. This is the cyst epithelial cells that line the cyst. And these are the cells that we're most interested in because those are the cells that give rise to the cyst and also secrete factors into the matrix adjacent to the cyst. And in this image here, I actually stained for a protein that I was interested in. It's called periostin. It's shows up as a brown product within the matrix, but it is secreted by the cyst epithelial cells. Um, and so we were able to provide either the blocks or tissue sections to investigators so they can study the proteins that they may be interested
Hey Darren,
in.
could
In
I ask
addition
a
to
quick question?
yeah, Julie, yeah, please.
Yeah, I'm just wondering how many people, how many kidneys are donated? Do you have any idea of how many people do that, like every year or every however often?
Yeah, that's a good question. That's something that I actually was hoping that we can have a discussion with the patient engagement group at some point. Um, Making patients aware of this opportunity has been difficult. The PKD Foundation sends out blast emails that kind of inform the patients about tissue donation. But and I also did a webinar at one point that you can go on the foundation's website and look it up to get additional information. But it'd be nice if we were able to make patients more aware of not only the opportunity, but also the importance of donating tissue for research. But
I
I
feel
would
like
say,
there are groups, like I feel like in the support groups, people want to do it. Like people talk about this when they're having a nephrectomy. And so I
yeah,
don't know if they just don't know how. So that would be, I would love to help with that in any way.
I appreciate that,
I
Julie.
can add that in May, when I underwent my bilateral nephrectomy, they actually, as part of the paperwork, you're being asked if you would like to donate. And
Mm,
I said yes, and I think most people do say yes. So it is actually brought to the patient.
yeah, it's one of the problems with the tissue donation process is that we need to have the kidneys fairly intact so that we can isolate the cells from the cyst. And a lot of the hospitals require that their pathology group section through the kidney to look for solid masses. And of course, there's a lot of variability in the pathology group on whether or not they're willing to work with us. Maybe have our pathologist do the path exam, or if maybe we can fix the kidneys after I isolate the tissue, and then ship the kidney back so that they can do the path exam. But one of the problems that we have, frequent problems, is being able to have the kidneys actually shipped to our university from the hospital without some type of analysis done to the kidney that kind of disrupts the architecture of the cystic kidney.
So Darren, you're actually describing an issue which is not related so much to the education of the patients, but something related to how different centers work. So who can actually help to solve that problem?
Well, I kind of wonder if, I mean, PKD Foundation has a consent form that the patient signs, but it might require a little bit more, consent form that gives the foundation permission to have the kidneys shipped, which alters the patient's normal care, and that gets a little bit complicated through the IRB process. But it's something that we can certainly discuss. Okay. Okay. So in addition to fixing the tissues for histology sections, we also freeze down tissue that can be thawed out, and investigators can look at protein levels or uh, you know changes in gene expression, et cetera. We also collect individual and pooled cyst fluids. A lot of times investigators want to look to see if proteins may be released from the cells and accumulate within the cyst fluid. And in some cases, we collect blood and urine at the time of nephrectomy, particularly if it's here at the Kansas University Hospital. And then this provides a really robust set of biomaterials for investigators. And I'm sorry, this is a little bit, maybe a little bit gross for some of the patients here, but this is a surface of a polycystic kidney, and this is how I isolate the cyst. After I isolate the cyst epithelium, I digest it and put it into a culture dish, and this is what the cells look like after they've proliferated and grown in culture. So what do we do with the PKD cells? We can put the cells in growth media that contains nutrients and salt that are very similar to body fluid. The cells are grown in incubators at 37 degrees Celsius, which is the same as body temperature, 98.6 degrees Fahrenheit. And with the similar oxygen and carbon dioxide content that mimics the environment inside the tissues. The incubators, this is actually an image of my cell culture laboratory, and these are the incubators that where we put the cells to grow. And this is what the inside of that incubator looks like. This would be a flask with cells growing on the inside of that flask. That the red appearance there is media that contains the nutrients and salt. It's red because it's got a pH indicator that allows us to know if the cells have consumed all their nutrients or now have started to secrete acid into the media, because we can see a change in the color of that media. So these cells can actually be immediately used for experiments. This would be an example where the cells are being used in different type of assays, or they can be lifted from the surface and spun down and frozen in vials, cryovials that contain media with something called DMSO. DMSO allows the cells to survive the freezing process by preventing crystals to form inside the cell as the liquid freezes. So these cells can be placed in these vials, frozen down, and then put in liquid nitrogen. Liquid nitrogen is very, very cold. It's below 300 degrees Fahrenheit. And this keeps the cells alive for decades. And that's really not an exaggeration. I've been doing this for over 30 years, and I can thaw out cells that I froze down 30 years ago and propagate them and use them in experiments. The nice thing is that we can also send these vials of cells to other laboratories where they can be thawed and propagated and used for in vitro assays at other institutions. Okay, so in my opinion, the 80 PKD cells are ideal for these type of in vitro assays for testing hypotheses and validating potential therapies. A lot of times we need to find some type of comparison for the disease state, and so we also collect cells from normal human kidney. These cells are typically from nephrectomy specimens. Say a patient has a tumor on one of the poles of the kidney and they have either a partial or complete nephrectomy and there's some normal tissue. We can grow normal human kidney cells from those tissues, but a lot of times we get kidneys from a transplant network here in Kansas City from kidneys that can't be transplanted for one reason or another, such as anatomical problems or vascular problems, or perhaps the kidney has slightly higher glomerular sclerosis than what would be accepted. Some of the common assays that we use these cells for is cell proliferation. That's shown down here on the left. This is a 96 well culture plate, and the cells are grown in each of these wells. So you might imagine that you can have 96 different drugs being tested at one time. I mean, obviously you would have replicates, but the point is that using these type of cell culture assays allows you to test a lot of compounds or test hypotheses using a large number of different conditions. The cells can also be grown on these permeable supports called transwells. On the permeable support means that it's able to, a fluid's able to flow through it. But as the cells grow on these supports, they form a monolayer, kind of like what lines the cyst wall. And a lot of the studies that I did when I was a postdoc and early faculty member was trying to understand how fluid accumulated within the cyst. And so we would do these fluid secretion assays using these transwells. One of the more popular assays nowadays is in vitro cyst growth, where the ADPKD cells can be seeded within a semi-solid matrix called collagen. And it's kind of like Jello, where it's semi-solid, but the cells can grow within it. And if you treat the cells with certain agonists, they'll form cysts, much like they do in vivo. And a lot of drug companies like to use those type of assays to test therapies. But other type of experiments that can be done is looking for changes in gene expression or protein abundance, or looking at pathways that might be involved in the signaling process for cyst growth. And you're all familiar with tolvaptan. Very early in the process of developing tolvaptan, Otsuka had contacted us to test tolvaptan against some of the other compounds that they had previously developed. And so we published this in 2011 before the clinical trial was published. But in this assay, we were using human ADPKD cells, and this is a cell proliferation experiment on the left. The control rate of cell proliferation was set to 100. And then you could see that when we treated with the hormone called vasopressin, we saw an increase in the proliferation rate by approximately 50%. And then if we added tolvaptan with increasing concentrations in the presence of the vasopressin, we saw inhibition of the cell proliferation, demonstrating that tolvaptan was effective on slowing the proliferation of the cystic epithelial cells. And so in addition to that, we looked at a pathway that we suspected was involved in the proliferation pathway, and found that tolvaptan also decreased the phosphorylation of this key mitogenic component in the signaling pathway. And then we also did in vitro cyst assay, where we treated with tolvaptan in the presence of vasopressin, and showed that it can inhibit cyst formation. And then, of course, that led to additional animal studies that were necessary for the clinical trial that ultimately was approved by the FDA. Okay, so one of the problems with using human ADPKD cells is that for comparison, we'd have to use normal human kidney cells from another patient. So genetically, they're quite different. And so that's always a concern, because it'd be better if we had what's considered to be an isogenic cell line. So it's the same genetic makeup for the control and the PKD cells. So let's say that we generate cell lines from a normal individual, and then simply introduce the PKD mutation. That allows us to make a better comparison on the effect of mutating that gene than making comparisons on two different genetic backgrounds. So one of the things that we want to do is establish cell lines, and these cell lines are considered to be immortal in that they can grow forever. The nice thing about cell lines compared to primary cells is they're easy to maintain. Most investigators can do that, and they're easy to distribute to other laboratories. So you don't have to be able to isolate the cyst epithelial cells. You simply have to thaw out the cells and grow them up. But to generate a cell line, you have to be able to immortalize the cells without changing their epithelial characteristics. Epithelial simply is the type of cell that lines the tubules and gives rise to the cyst. So to generate an isogenic line, we need to be able to introduce an approach to cause the cells to switch from being normal to PKD. And this in itself is a tricky process of genetic engineering. So Owen Woodwork at University of Maryland, Baltimore has been very successful in generating isogenic mouse cell lines that are normal in PKD. And how he did with this was to collaborate with the Animal Corps at University of Maryland, where they obtained a mouse called the Immorta mouse. This mouse expresses SV40. This is the simian virus large T antigen, and this immortalizes cells. And so they bred this Immorta mouse into some of their PKD cell lines that were inducible. I think Dr. Parnell probably talked about mice that you can induce the PKD phenotype after they are established. So by making these cells immortal, Dr. Woodward is able to isolate the kidneys, digest them to single cell, propagate the cells, and then establish clones from single cells that are isolated from the culture. And since they're immortal, single cells will give rise to clones, and then you can characterize the various clones. And since these are an inducible line for knocking out PKD1 or PKD2, all he has to do is use this compound called doxycycline, which will make normal kidney cells into PKD cells. And this is just kind of confirmation where he was able to run the proteins from these lysates. And this band here that you're looking at is polycystin 2, the gene product from the PKD gene. And when he added doxycycline, you could see that there's a loss of the polycystin 2 protein. Then after taking these cells and knocking out PKD2, he's able to put these cells in the collagen matrix and they form cysts.
Now, just a quick question, since you mentioned doxycycline. So in addition to looking for potential treatments, do you, for any groups looking actually using the same technique to find out what commonly prescribed medication or other related chemicals actually promote the PKD or speed
Yeah,
it
so the
up?
doxycycline, I'm sorry, the doxycycline is actually a tool for activating an enzyme that removes PKD2. It's not just a random compound. And I'm not sure if there's other, I mean, another system would be using tamoxifen, but that has some complications associated with it. But the system that they've designed is for doxycycline to activate the Cre recombinase that removes the gene. Does that answer your question, Sari?
Partially, the flip side of it would be whether anybody is looking for compounds but actually not removing the gene or maybe speeding up the proliferation or causing harm as opposed to benefit.
Yeah. Yeah, I don't know if I can answer that. So it sounds to me like you're asking if there's compounds that would spontaneously cause normal cells to become cystic cells without the gene manipulation. Is that what you're asking me?
Or if you have already the gene manipulation, do you ever experiment with compounds which, instead of slowing the progression of proliferation, it's actually speeded up?
Yeah. I don't know the answer to that. I don't know if there's compounds that actually accelerate, intentionally to accelerate the cyst formation after you've genetically knocked out PKD1 with doxycycline. So yeah, so Owen Woodward has generated a panel of PKD1 mouse cell lines, both from male and female mice and from different regions within the kidney. So he's in the process of characterizing a variety of different clones from this method of generating the isogenic PKD1 and wild type cells. In addition to that, he's using a kind of a clever technique where he can label PC2 in red. So this red structure here means that there's normal polycystin 2 expression. And what he found is that those cells that are expressing polycystin 2, when you put them in collagen, they actually have the capability of forming tubule-like structures. But then if you add the doxycycline and knock out the PKD2 gene, the cells become green. And these cells actually form cysts. So this will be a nice tool for investigators to look to see if the loss of polycystin 2 gives a particular phenotype. And even in a culture where there may not be 100% knockout of the PKD2, they can identify which cell structures have formed from cells with and without the PKD2 gene. Ultimately, what we'd like to do is generate isogenic human normal and PKD cell lines. Of course, this is a little bit more challenging in that, the cells are going to have to be isolated from individuals, and then we have to immortalize the cells without affecting their characteristics. And so UAB and Kansas have both been working on this. And we've immortalized cells using a temperature-sensitive SV40. And what that means is that the cells that are expressing the temperature SV40, they'll grow at a lower temperature in the incubator at 33 degrees. That causes the SV40 to be expressed, driving the proliferation of the cells. But then we can move the cells to 37 degrees, and that shuts off the expression of the SV40 large T antigen, and these cells begin to behave normally as far as their growth characteristics. One of the concerns about using this method is that if you have too many copies of the temperature sensitive SV40, this could cause cells to proliferate too rapidly, and then they take on a de-differentiated phenotype because you've altered their characteristics somehow. So what we're working on is different methods of immortalization of the cells, and what we're working on is a human telomerase. And this is a little bit different. Classical immortalization typically uses oncogenes, and this inevitably gives rise to cells that have cancer-associated changes, genomic instability, their cell cycle checkpoints that control their cell proliferation may be disrupted, and they may not be fully capable of full cellular differentiation. So what we're hoping is that the hTERT will allow us to immortalize cells without affecting some of these cellular characteristics. Okay, so another approach would be to use pluripotent stem cells. And you may have heard of embryonic stem cells. These are cells derived from the inner cell mass of the blastocyst. This is an early stage pre-implanted embryo. These cells are pluripotent, meaning that they can give rise to a bunch of different cell types and structures. They undergo cell renewal indefinitely, meaning that they will continue to proliferate in a de-differentiated state. So you could distribute these cells to multiple investigators, and then they can induce them to form different types of tissues. However, the use of human embryonic stem cells is controversial. And so investigators have come up with a way of taking cells from biopsies, either skin or blood, and reprogramming those cells to become stem cells. And so in this process, this reprogramming of the cells is a very sophisticated approach where you have to treat the cells with different concentrations of factors. It causes the cells to express certain things, but it's also temporal. So it's not an easy process of inducing these iPS cells. But once they become iPS cells, they resemble embryonic stem cells. They have self-renewal. They will differentiate into a variety of different cell types and structures. In this image, they're actually proposing to do gene editing to correct the gene mutation and reintroduce it back into the patient. That's not exactly what I don't think we're quite ready for anything like that, but we can use these cells to differentiate them into kidney-like structures, and then look to see how mutations in the polycystins affect these structures that form, particularly called organoids. And so John Perrant at UAB is leading the process of taking normal iPS cells and using CRISPR gene editing to knock out PKD1 and PKD2 and other genes, with the idea that these cells will be used for forming these organoids. So early on, when I was talking about some of the limitations of using in vitro assays, is that they don't fully represent the complexity of the in vivo situation, but these organoids are capable of forming a variety of different structures, different tubule types. They have similarities to glomeruli, interstitial cells. So this actually may be a useful tool for looking at some of the crosstalk between different cell types when you introduce PKD1 mutations. I've listed some of the applications for using these iPS cells, including the formation of organoids and evaluating cyst formation. So down here is some work from Jack Song at UAB, where he took normal cells and formed an organoid. It's not lit up like the other one. It's not fluorescent. But what he found is that if you knock out PKD2, you can get cyst formation in these organoids. And this had been demonstrated previously by Dino Friedman's group using iPS cells. And over on the left is wild type cells, and then on the right is PKD mutant cells. And he actually was able to demonstrate that these cells have the capability of forming quite large cysts in culture. And so a lot of people are interested in using these organoids to test different hypotheses and perhaps even therapeutic agents. And so John Perrant at UAB has been in the process of generating a lot of iPS cell lines and making them available to investigators. And this just kind of lists the various mutations that he's introduced. This would be a heterozygous. This would be kind of like what patients inherit, where only one allele is mutated. But a lot of people are interested in PKD1 null, PKD2 null cell lines. He introduced mutations into PKHD1. This is the gene that's responsible for AR-PKD. He's also introduced mutations into an IFT88. This is a gene that's involved in cilia formation, and we know if we mutate the cilia that's located on the cell, you can also get cyst formation. More sophisticated alterations he's done is he's introduced a patient mutation into PKD2. It's also got conditional knockout and conditional restore PKD2 cell lines. And he's also generated a PKD2 halo, which I think Dr. Parnell may have talked a little bit in the animal subcommittee, where they've tagged the polycystin 2 protein. And so he's introduced that tag into the iPS cells. What's in development is that he's now got human embryonic stem cells. The NIH has a cell line that we're able to use and distribute. And he's in the process of introducing these mutations into the true embryonic stem cells. Okay, in addition to that, we have a number of cell lines. I just listed a few here that we've obtained from either other subcommittees. I mentioned the halo cells. We have a repository of those cells. We also have the PKD1 SNAP cells that Dr. Parnell had generated in the in vivo subcommittee. But we also have cells that were provided from other investigators. And so we kind of serve as a repository for a variety of cells to distribute to the community. And finally, the subcommittee does a lot of optimization of cell assays, and we also provide training to investigators on how to perform these assays. And these protocols are posted on the website for both the maintenance of growing and propagating the primary and immortalized cells, but also for some of the in vivo assays, including that optimized protocol for growing human ADPKD cells and in vitro cyst assays. So this is the subcommittee members. Paul Welling is co-chair of the subcommittee. He's at Johns Hopkins. I talked a little bit about Owen Woodward's work at Maryland and John Perront's work at UAB. But Courtney and Jack and Medulik are also members of the subcommittee and are involved in different aspects of it. So hopefully that was useful to you guys, and I'd be happy to try to answer any questions you might have.
I have a question. I probably won't say it very well, but okay. So my understanding is that you make the cells by inducing PKD into them, so it's not naturally occurring. Have there been any studies that compare the cells you make to a naturally occurring PKD, like me? You know, if I gave my cells, and to see that if you're introducing the compound, it behaves similarly in both groups?
Well, you know, I mean, in the 80 PKD cells that we isolate from patients, those would have whatever genetic mutation that initiated cyst formation, right? So, but you're talking about cell lines where we may introduce a particular mutation that's known to cause the disease in patients. And actually, we did that with the iPS cells. But I mean, you know, I think that the idea, I think our long-term goal would be to have a way to introduce particular mutations that have been shown to be pathogenic in patients for in vitro assays. Right now, it's not convenient to introduce a large panel of different mutations because we kind of have to start from the beginning. But you know, I think some of the engineering, genetic engineering that they're trying to do is so that we can switch in different mutations conveniently so that we can study that particular type of mutation in vitro assays.
How I can add or maybe expand or clarify Susan's question. I think that what we are thinking about, and I was about to ask similar question as Susan's, is we know from other types of diseases, possibly where you grow cells, you test things on the cells, which present or represent similar, all the what needs to manifest a disease of some sort. And yet, when you try it on a human, a whole human, where those cells are part of a complicated system, sometimes the medication or the drug possibly won't work as it worked in the Petri dish, even though those cells came from human with the disease or it was induced. So I think the question is maybe two points. One is, do you compare how those effects manifest themselves on your Petri dish, so to speak, versus what happened in a real human with the disease? And second, do you have explanation or ideas about what cause sometimes something work in the Petri dish and does not work in a full human?
Yeah, that's a good point. It's hard to make a direct comparison about the impact of a drug in an in vitro assay in human patients because, with the in vitro assays, we're looking at some type of readout. We may treat the cells with a drug and look at particular pathways that we think would be affected by the drug. And it's kind of hard to do that, dose a patient with a compound and then be able to isolate tissue from that patient to see if it impacts the pathway in situ, in vivo. However, we've had some discussion with pharmaceutical companies that would like to do a study where we know that the patient's going to have a nephrectomy, so they're in stage, and then perhaps consent the patient so that they receive a compound. And then after the kidney is removed, we look to see if it impacted the pathway within the kidneys. But, all that is, that's really going to have to be approved by institution review board. And there has to be some type of level of safety on the drug that's being tested. So if they've already done like a phase 1 trial with a compound and we know it's safe in patients, then dosing it in a patient that's going to have a nephrectomy might be a possibility to see if it has a similar impact in the tissue as it did in an in vitro assay or in an animal model.
Yeah, and we can all probably understand how complicated it is and sometimes impossible to do it on real human, as you said, before the drug is proven to at least not kill the patient. But what about using, I don't want to say necessarily AI, but deep machine learning on all the huge amount of data we have, where you compare the whole process and results in, take for example, Tavaptan, which is, I'm assuming there are much data these days about it. Has anybody looked into actually doing data analysis comparing what happened in the Petri dish to what happened in people? For example, we know it slows down the disease in people, but does it slow down in the same rate as it is in the Petri dish? Example of one parameter, I'm sure there are many more parameters.
Yeah, that's a good question. I'm not entirely sure I can answer that. I do know that there's some people that are doing RNA-seq analysis. So RNA-seq would be looking at changes in transcription of different genes. And by doing that, you can kind of get an idea of what pathways in the cell are affected. And then they can treat cells with a drug and see if those pathways that they identified in disease might be affected by the drug and reverse that. So that, I mean, obviously it's not exactly what you're asking, but they may be using some of the large data sets to determine whether or not a drug has the potential for correcting some of the pathways that are defective in a PKD patient or PKD tissue. But I don't know about, whether or not machine learning. Maybe Lisa can touch on that if she's
Yeah, so
has any knowledge about it.
I think that that one of the things that is coming to kidney disease is exactly what you're talking about, Sarah, is the idea of using various AI tools to model. And that can be everything from knowledge graphs, where you take a whole bunch of inputs, big data around a whole bunch of inputs, and then you ask the computer to make relationships. And they may be relationships that humans just don't know about. So knowledge graph approaches is coming to kidney disease. There are trying to understand some of the genetic mechanisms that might be going on. There are strategies that are new strategies that are looking to see not just, is there an association between a particular address in the genetic information, but within those addresses, are those regions of the genome, are they kind of open for business? In other words, can genes in that region be expressed? And if they are they expressed in the kidney? And so there's a whole new level of understanding. So knowledge graphs to come up with pathways, various strategies to sort of ask deep questions about not just, this particular neighborhood has genes that might be interested in modifying the disease, but really be able to drill down to go, okay, we got a whole neighborhood here. We got a city block of houses. Which house is it? And there are strategies to do that. There also I think, are ways that people are looking at clinical data. These are called PheWAS studies, Phe standing for phenotype or clinical presentation, and marrying it with genetic information to begin to get a deeper understanding of what you guys all know. Not everybody with PKD has the same disease. Not everybody with PKD has the same clinical course. And so what are the genetic factors that differentiate these genetic courses? So these kinds of tools are being applied in other fields, and I think that increasingly these tools are coming to PKD. For example, just to the final thing that I'll say, we have our big FASB meeting on PKD coming up next week. And there are people who are leading multidisciplinary teams that involve not just biologists, but biophysicists and computer scientists, and trying to take that complementary set of expertise to take fresh looks at some of the problems that Darren is talking about. But I think a fundamentally important point that Darren, I think, made really well is that we need to have material from which we can generate new data. So it's not just data that exists, but new data when we have some perspective to go, oh, okay, let's do this, and this to generate new data sets to test the hypotheses that are coming forward. So I do think AI is coming in a big way to kidney-related research and certainly to PKD.
That's
Did
good
that
to
help?
hear. Yeah, actually, you answered some of my other questions about indeed using the huge data to understand the difference of progression of disease between people what seemingly seem the same. But
Right.
and it's done so nicely for, I think some people heard about the deep machine learning used for finding new antibiotics, because we cannot do all the connection and finding that some particle in earth, some type of fungi or whatever in soil can actually work as an antibiotic. So that's good to hear. Sign me in if anybody needs somebody to.
Right. Well, and I think that one of the other things is, remember, it's not just genetic. Environment plays a role. And some of the things that AI is beginning to allow us to capture in a manageable way is, what are these AI factors? Everything, I mean, our biggest exposure is what we eat and what we drink. Well, what does that mean in a granular way? Because if we knew, for example, we used to think, and unfortunately it's been debunked, but we used to think caffeine accelerated cyst progression in ADPKD. But wouldn't it be good if we really were able to understand, you've got PKD, you're 10 years old. Avoid this, this, and this, because we know exactly the pathways that are getting triggered.
Yeah.
We're not there yet, but people are beginning to ask those questions.
It's good to hear that deep machine learning find a place now in PKD. Great.
So, Lisa, do you think there needs to be more epigenetic type of studies then?
Absolutely. I mean, so what Darren is talking about was the thing I kind of alluded to. Remember, DNA isn't just a string. It's not just a string of pearls. The pearls they twist and they bend. And if you think about, mostly women on this call, sorry, Darren, if you think about complicated jewelry from like the Gilded Age, you had the twists of pearls, and then some of the pearls got caught up in some other metal or gems or this, that, the other thing. Is all very too fancy. But that's the way DNA is. And if you're going to make a protein, you got to open up that pearl string and just lay it out so that it can make the protein from all those beads. What Darren's talking about with epigenetics is the way that whole big mess of DNA gets folded in a very clever way is by a series of proteins, and the core of which are called histones. And so they have to release the DNA. They have to release the pearl string to open it up. And that's not just something that's done in every cell of the body in the same way. It's done in different cells. So that's when I was talking about marrying up all this information, not just the string of pearl information, but then where in the kidney is that string of pearls open? And what are those genes that string of pearls is going to make? That's important. When we think about a whole nother genome, the mitochondria, who would have ever thought that mitochondria, these little energy packets in the cell, and we got hundreds of them in every cell, was going to be important in PKD, but Alessandro Boletta and Greg Germino and others have shown that it is. And so all of this is an incredible amount of data. How do we put it all together? AI is going to help us do that. And so bringing computer scientists into the field is really, really important. I know, for example, in the UAB grant, one of the things that we built into our grant was a whole core on informatics, computer science expertise to begin to start to do this. And I'll bet you the other sites are going to do this as well. I mean, I can't wait to see who the funding folks are going to decide is going to be able to go forward. I hope it's this core and maybe some additional groups. And I hope in both this core and the addition, computer scientists are going to figure prominently in our next iteration. So I hope that helped add some color commentary, Darren.
It did. Thank you, Lisa.
Okay, guys, it's 8:00 in the East, and I'm hungry, so I gotta go.
Darren, thanks so much for coming.
Yes,
Awesome.
thank you very much. It's very interesting.
Thanks, Darren.
Appreciate
So
it.
long.
Bye-bye.